Crypsis. In ecology, crypsis is the ability of an animal or a plant 1. To avoid observation or detection by other animals. It may be a predation strategy or an anti-predator adaptation. Methods include camouflage, nocturnality, subterranean lifestyle and mimicry. Crypsis can involve visual, olfactory with pheromones or auditory concealment. When it is visual, the term cryptic coloration, effectively a synonym for animal camouflage, is sometimes used, but many different methods of camouflage are employed in nature. Hiding. Revealing itself. Cryptic behavior. Mossy leaf-tailed gecko, Uroplatus sicerae, Montagne d'Amber, Madagascar, showing camouflage using a flange of skin to eliminate shadow. Overview. There is a strong evolutionary pressure for prey animals to avoid predators through camouflage, and for predators to be able to detect camouflaged prey. There can be a self-perpetuating coevolution, in the shape of an evolutionary arms race, between the perceptive abilities of animals attempting to detect the cryptic animal and the cryptic characteristics of the hiding species. 2. Methods. A Draco lizard showing camouflage methods including background matching, disruptive coloration, reduction of shadow, and cryptic behavior in Bandipur National Park. Methods of crypsis include visual camouflage, nocturnality, and subterranean lifestyle. Camouflage can be achieved by a wide variety of methods, from disruptive coloration to transparency and some forms of mimicry, even in habitats like the open sea where there is no background. 3. 4. As a strategy, crypsis is used by predators against prey and by prey against predators. 3. Crypsis also applies to eggs. 5. And pheromone production. 6. Crypsis can in principle involve visual, olfactory, or auditory camouflage. 7. Visual. Camouflage allows animals like this disruptively patterned spider to capture prey more easily. Many animals have evolved so that they visually resemble their surroundings by using any of the many methods of natural camouflage that may match the color and texture of the surroundings, cryptic coloration, and or break up the visual outline of the animal itself, disruptive coloration. Such animals, like the tawny dragon lizard, may resemble rocks, sand, twigs, leaves, and even bird droppings, mimesis. Other methods including transparency and silvering are widely used by marine animals. 8. Some animals change color in changing environments seasonally, as in ermine and snowshoe hare, or far more rapidly with chromatophores in their integuments, as in chameleon and cephalopods such as squid. Countershading, the use of different colors on upper and lower surfaces in graduating tones from a light belly to a darker back, is common in the sea and on land. It is sometimes called Thayer's Law, after the American artist Abbott Henderson Thayer, who published a paper on the form in 1896 that explained that countershading paints out shadows to make solid objects appear flat, reversing the way that artists use paint to make flat paintings contain solid objects. Where the background is brighter than is possible even with white pigment, counter-illumination in marine animals, such as squid, can use light to match the background. Some animals actively camouflage themselves with local materials. The decorator crabs attach plants, animals, small stones, or shell fragments to their carapaces to provide camouflage that matches the local environment. Some species preferentially select stinging animals such as sea anemones or noxious plants, benefiting from aposematism as well as are instead of crypsis. 9. Olfactory. Some animals, in both terrestrial and aquatic environments, appear to camouflage their odor, which might otherwise attract predators. 10. Numerous arthropods, both insects and spiders, mimic ants, whether to avoid predation, to hunt ants, or, as in the large blue butterfly caterpillar, to trick the ants into feeding them. 11. Pirate perch, Aphrododorus cyanus, may exhibit chemical crypsis, making them undetectable to frogs and insects colonizing ponds. 12. Trained dogs and meerkats, both scent-oriented predators, have been shown to have difficulty detecting puff adders, whose strategy of ambushing prey necessitates concealment from both predators and prey. 13. Auditory. Some insects, notably some noctuid moths, such as the large yellow underwing, and some tiger moths, such as the garden tiger, have been supposed to defend themselves against predation by echolocating bats, both by passively absorbing sound with soft, fur-like body coverings and by actively creating sounds to mimic echoes from other locations or objects. The active strategy was described as a phantom echo, that might therefore represent auditory crypsis, with alternative theories about interfering with the bat's echolocation, jamming. 14. 15. Subsequent research has provided evidence for only 2 functions of moth sounds, neither of which involve auditory crypsis. Tiger moth species appear to cluster into 2 distinct groups. One type produces sounds as acoustic aposematism, warning the bats that the moths are unpalatable. 16. Or at least performing as acoustic mimics of unpalatable moths. 17. The other type uses sonar jamming. In the latter type of moth, detailed analyses failed to support a phantom echo mechanism underlying sonar jamming, but instead pointed towards echo interference. Genetic variation. Genetic variation is the difference in DNA among individuals. 1. Or the differences between populations among the same species. 2. The multiple sources of genetic variation include mutation and genetic recombination. 3. Mutations are the ultimate sources of genetic variation, but other mechanisms, such as genetic drift, contribute to it, as well. 2. Darwin's finches are Galapagos finches. 4. Parents have similar gene coding in this specific situation where they reproduce and variation in the offspring is seen. Offspring containing the variation also reproduce and passes down traits to their offspring. Among individuals within a population, genetic variation can be identified at many levels. Identifying genetic variation is possible from observations of phenotypic variation in either quantitative traits, traits that vary continuously and are coded for by many genes, e.g., leg length in dogs, or discrete traits, traits that fall into discrete categories and are coded for by one or a few genes, e.g., white, pink, or red petal color in certain flowers. Genetic variation can also be identified by examining variation at the level of enzymes using the process of protein electrophoresis. 5 polymorphic genes have more than one allele at each locus. Half of the genes that code for enzymes in insects and plants may be polymorphic, whereas polymorphisms are less common among vertebrates. Ultimately, genetic variation is caused by variation in the order of bases in the nucleotides in genes. New technology now allows scientists to directly sequence DNA, which has identified even more genetic variation than was previously detected by protein electrophoresis. Examination of DNA has shown genetic variation in both coding regions and in the non-coding intron region of genes. Genetic variation will result in phenotypic variation if variation in the order of nucleotides in the DNA sequence results in a difference in the order of amino acids in proteins coded by that DNA sequence, and if the resultant differences in amino acid sequence influence the shape, and thus the function of the enzyme. 6. Between populations. Differences between populations resulting from geographic separation is known as geographic variation. Natural selection, genetic drift, and gene flow can all contribute to geographic variation. 7. Measurement. Genetic variation within a population is commonly measured as the percentage of polymorphic gene loci or the percentage of gene loci in heterozygous individuals. The results can be very useful in understanding the process of adaptation to the environment of each individual in the population. 8. Sources. A range of variability in the Musseldonax variabilis. Random mutations are the ultimate source of genetic variation. Mutations are likely to be rare, and most mutations are neutral or deleterious, but in some instances, the new alleles can be favored by natural selection. Polyploidy is an example of chromosomal mutation. Polyploidy is a condition wherein organisms have 3 or more sets of genetic variation, 3n or more. Crossing over, genetic recombination, and random segregation during meiosis can result in the production of new alleles or new combinations of alleles. Furthermore, random fertilization also contributes to variation. Variation and recombination can be facilitated by transposable genetic elements, endogenous retroviruses, lines, signs, etc. For a given genome of a multicellular organism, genetic variation may be acquired in somatic cells or inherited through the germline. Forms. Genetic variation can be divided into different forms according to the size and type of genomic variation underpinning genetic change. Small-scale sequence variation, less than 1 kilobase, kb, includes base pair substitution and indels. 9. Large-scale structural variation, greater than 1 kilobit, can be either copy number variation, loss or gain, or chromosomal rearrangement, translocation, inversion, or segmental acquired uniparental disomy. 9. Genetic variation and recombination by transposable elements and endogenous retroviruses sometimes is supplemented by a variety of persistent viruses and their defectives which generate genetic novelty in host genomes. Numerical variation in whole chromosomes or genomes can be either polyploidy or aneuploidy. Maintenance in populations. A variety of factors maintain genetic variation in populations. Potentially harmful recessive alleles can be hidden from selection in the heterozygous individuals. In populations of diploid organisms, recessive alleles are only expressed in the less common homozygous individuals. Natural selection can also maintain genetic variation in balanced polymorphisms. Balanced polymorphisms may occur when heterozygotes are favored or when selection is frequency dependent. RNA viruses. A high mutation rate caused by the lack of a proofreading mechanism appears to be a major source of the genetic variation that contributes to RNA virus evolution. 10. Genetic recombination also has been shown to play a key role in generating the genetic variation that underlies RNA virus evolution. 10. Numerous RNA viruses are capable of genetic recombination when at least 2 viral genomes are present in the same host cell. 11. RNA recombination appears to be a major driving force in determining genome architecture and the course of viral evolution among picornaviridae plus ssRNA e.g. poliovirus. 12. In the retroviridae plus ssRNA e.g. HIV. Damage in the RNA genome appears to be avoided during reverse transcription by strand switching, a form of genetic recombination. 13, 14, 15. Recombination also occurs in the coronaviridae plus ssRNA e.g. SARS. 16. Recombination in RNA viruses appears to be an adaptation for coping with genome damage. 11. Recombination can occur infrequently between animal viruses of the same species but of divergent lineages. The resulting recombinant viruses may sometimes cause an outbreak of infection in humans. 16. History of genetic variation. Evolutionary biologists are often concerned with genetic variation, a term which in modern times has come to refer to differences in DNA sequences among individuals. However, quantifying and understanding genetic variation has been a central aim of those interested in understanding the varied life on Earth since long before the sequencing of the first full genome, and even before the discovery of DNA as the molecule responsible for heredity. While today's definition of genetic variation relies on contemporary molecular genetics, the idea of heritable variation was of central importance to those interested in the substance and development of life even before the writings of Charles Darwin. The concept of heritable variation, the presence of innate differences between life forms that are passed from parents to offspring, especially within categories such as species, does not rely on modern ideas of genetics, which were unavailable to 18th and 19th century minds. Pre-Darwinian concepts of heritable variation. In the mid-1700s, Pierre-Louis Maupertuis, a French scholar now known primarily for his work in mathematics and physics, posited that while species have a true, original form, accidents during the development of nascent offspring could introduce variations that could accumulate over time. 17. In his 1750 Essai de Cosmologie, he proposed that the species we see today are only a small fraction of the many variations produced by a blind destiny, and that many of these variations did not conform to their needs, thus did not survive. 18. In fact, some historians even suggest that his ideas anticipated the laws of inheritance further developed by Gregor Mendel. 19. Simultaneously, French philosopher Denis Diderot proposed a different framework for the generation of heritable variation. Diderot borrowed Maupertuis's idea that variation could be introduced during reproduction and the subsequent growth of offspring. 20. And thought that production of a normal organism was no more probable than production of a monstrous one. 21. However, Diderot also believed that matter itself had lifelike properties and could self-assemble into structures with the potential for life. 20. Diderot's ideas on biological transformation, introduced in his 1749 work Letter on the Blind, were thus focused on variability of spontaneously generated forms, not variability within existing species. 22. Both Maupertuis and Diderot built on the ideas of Roman poet and philosopher Lucretius, who wrote in De Rerum Natura that all the universe was created by random chance, and only the beings that were not self-contradictory survived. 23. Maupertuis's work is distinguished from the work of both Lucretius and Diderot in his use of the concept of conformity in explaining differential survival of beings, a new idea among those who believe that life changed over time. 23. Like Diderot, two other influential minds of the 18th century, Erasmus Darwin and Jean-Baptiste Lamarck, believed that only very simple organisms could be generated by spontaneous generation, so another mechanism was necessary to generate the great variability of complex life observed on Earth. 17. Erasmus Darwin proposed that changes acquired during an animal's life could be passed to its offspring, and that these changes seemed to be produced by the animal's endeavors to meet its basic needs. 24. Similarly, Lamarck's theory of the variability among living things was rooted in patterns of use and disuse, which he believed led to heritable physiological changes. 17. Both Erasmus Darwin and Lamarck believed that variation, whether it arose during development or during the animal's life, was heritable, a key step in theories of change over time extending from individuals to populations. In the subsequent century, William Herschel's telescopic observations of diverse nebulae across the night sky suggested to him that different nebulae could each be in different stages in the process of condensation. This idea, which came to be known as the nebular hypothesis, suggested that natural processes could both create order out of matter and introduce variation, and that these processes could be observed over time. 17. While it may seem to the modern reader that astronomical theories are irrelevant to theories of organic variation, these ideas became significantly conflated with ideas of biological transformation, what we now know as evolution, in the mid-19th century, laying important groundwork for the work of subsequent thinkers such as Charles Darwin. 25. Darwin's concept of heritable variation. Charles Darwin's ideas of heritable variation were shaped by both his own scientific work and the ideas of his contemporaries and predecessors. 26. Darwin ascribed heritable variation to many factors, but particularly emphasized environmental forces acting on the body. His theory of inheritance was rooted in the now disproven idea of gemmules, small, hypothetical particles, which capture the essence of an organism and travel from all over the body to the reproductive organs, from which they are passed to offspring. 27. Darwin believed that the causal relationship between the environment and the body was so complex that the variation this relationship produced was inherently unpredictable. 28. However, like Lamarck, he acknowledged that variability could also be introduced by patterns of use and disuse of organs. 29. Darwin was fascinated by variation in both natural and domesticated populations, and his realization that individuals in a population exhibited seemingly purposeless variation was largely driven by his experiences working with animal breeders. 30. Darwin believed that species changed gradually, through the accumulation of small, continuous variations, a concept that would remain hotly contested into the 20th century. 31. Post-Darwinian concepts of heritable variation. In the 20th century, a field that came to be known as population genetics developed. This field seeks to understand and quantify genetic variation. 31. The section below consists of a timeline of selected developments in population genetics, with a focus on methods for quantifying genetic variation. 1866 Heterozygosity. Gregor Mendel's hybridization experiments introduced the concept that in the 1950s came to be recognized as heterozygosity. 29. In a diploid species, one that contains two copies of DNA within each cell, one from each parent, an individual is said to be a heterozygote at a particular location in the genome if its two copies of DNA differ at that site. Heterozygosity, the average frequency of heterozygotes in a population, became a fundamental measure of the genetic variation in a population by the mid-20th century. 32. If the heterozygosity of a population is zero, every individual is homozygous, that is, every individual has two copies of the same allele at the locus of interest and no genetic variation exists. 1918 Variance. In a seminal paper entitled, The Correlation Between Relatives on the Supposition of Mendelian Inheritance, R.A. Fisher introduced the statistical concept of variance, the average of squared deviations of a collection of observations from their mean. Whereas the variance n is the mean of the population from which the observations are drawn. 33. R. Fisher's work in population genetics was not just important to population genetics, these ideas would also form the foundations of modern statistics. 1918, 1921 Additive and dominant genetic variance. R. A. Fisher subsequently subdivided his General definition of variance into two components relevant to population genetics, additive and dominant genetic variance. 34. An additive genetic model assumes that genes do not interact if the number of the genes affecting the phenotype is small and that a trait value can be estimated simply by summing the effect of each gene on the trait. Under Fisher's model, the total genetic variance is the sum of the additive genetic variance, the variance in a trait due to these additive effects, and the dominant genetic variance which accounts for interactions between genes. 33. 1948 Entropy. 1948 Entropy. Unlike variance, which was developed with the purpose of quantifying genetic variance, Claude Shannon's measure of diversity, now known as Shannon entropy, was developed as part of his work in communication theory as a way to quantify the amount of information contained in a message. However, the method quickly found use in population genetics, and was the central method used to quantify genetic diversity in a seminal paper by Richard Lewontin, The Apportionment of Human Genetic Diversity. 35. 1951 F statistics. F statistics, also known as fixation indices, were developed by population geneticist Sewall Wright to quantify differences in genetic variation within and between populations. The most common of these statistics, FST, considers in its simplest definition two different versions of a gene, or alleles, and two populations that contain one or both of these two alleles. FST quantifies the genetic variability among these two populations by computing the average frequency of heterozygotes across the two populations relative to the frequency of heterozygotes if the two populations were pooled. 36. F statistics introduced the idea of quantifying hierarchical concepts of variance and would become the foundation of many important population genetic methods, including a set of methods that tests for evidence of natural selection in the genome. Population bottleneck. A population bottleneck or genetic bottleneck is a sharp reduction in the size of a population due to environmental events such as famines, earthquakes, floods, fires, disease, and droughts, or human activities such as genocide, specioside, widespread violence or intentional culling. Such events can reduce the variation in the gene pool of a population. Thereafter, a smaller population, with a smaller genetic diversity, remains to pass on genes to future generations of offspring. Genetic diversity remains lower, increasing only when gene flow from another population occurs or very slowly increasing with time as random mutations occur. 1. This results in a reduction in the robustness of the population and in its ability to adapt to and survive selecting environmental changes, such as climate change or a shift in available resources. 2. Alternatively, if survivors of the bottleneck are the individuals with the greatest genetic fitness, the frequency of the fitter genes within the gene pool is increased, while the pool itself is reduced. Population bottleneck followed by recovery or extinction. The genetic drift caused by a population bottleneck can change the proportional random distribution of alleles and even lead to loss of alleles. The chances of inbreeding and genetic homogeneity can increase, possibly leading to inbreeding depression. Smaller population size can also cause deleterious mutations to accumulate. 3. Population bottlenecks play an important role in conservation biology, see minimum viable population size, and in the context of agriculture, biological and pest control. 4. Minimum viable population size. In conservation biology, minimum viable population (MVP) size helps to determine the effective population size when a population is at risk for extinction. 5. 6. The effects of a population bottleneck often depend on the number of individuals remaining after the bottleneck and how that compares to the minimum viable population size. Founder effects. A slightly different form of bottleneck can occur if a small group becomes reproductively, e.g., geographically, separated from the main population, such as through a founder event, e.g., if a few members of a species successfully colonize a new isolated island, or from small captive breeding programs such as animals at a zoo. Alternatively, invasive species can undergo population bottlenecks through founder events when introduced into their invaded range. 7. Examples. Humans. According to a 1999 model, a severe population bottleneck, or more specifically a full-fledged speciation, occurred among a group of Australopithecina as they transitioned into the species known as Homo erectus 2 million years ago. It is believed that additional bottlenecks must have occurred since Homo erectus started walking the earth, but current archaeological, paleontological, and genetic data are inadequate to give much reliable information about such conjectured bottlenecks. 8. Nonetheless, a 2023 genetic analysis discerned such a human ancestor population bottleneck of a possible 100,000 to 1,000 individuals, around 930,000 and 813,000 years ago, which lasted for about 117,000 years and brought human ancestors close to extinction. 9. 10. A 2005 study from Rutgers University theorized that the pre-1492 native populations of the Americas are the descendants of only 70 individuals who crossed the land bridge between Asia and North America. 11. The Neolithic Y chromosome bottleneck refers to a period around 5000 BC where the diversity in the male Y chromosome dropped precipitously. The Neolithic Y chromosome bottleneck refers to a period around 5000 BC where the diversity in the male Y chromosome dropped precipitously, to a level equivalent to reproduction occurring with a ratio between men and women of 1:17. 12. Discovered in 2015-13, the research suggests that the reason for the bottleneck was not a reduction in the number of males, but a drastic decrease in the percentage of males with reproductive success. Toba catastrophe theory. The controversial Toba catastrophe theory, presented in the late 1990s to early 2000s, suggested that a bottleneck of the human population occurred approximately 75,000 years ago, proposing that year American bison est The human population was reduced to perhaps 10,000 30,000 individuals. 14. When the Toba supervolcano in Indonesia erupted and triggered a major environmental change. Parallel bottlenecks were proposed to exist among chimpanzees, gorillas, rhesus macaques, orangutans and tigers. 15. The hypothesis was based on geological evidence of sudden climate change and on coalescence evidence of some genes, including mitochondrial DNA, Y chromosome DNA and some nuclear genes. 16. And the relatively low level of genetic variation in humans. 14. However, subsequent research, especially in the 2010s, appeared to refute both the climate argument and the genetic argument. Recent research shows the extent of climate change was much smaller than believed by proponents of the theory. 17. In 2000, a molecular biology and evolution paper suggested a transplanting model or a long bottleneck, to account for the limited genetic variation, rather than a catastrophic environmental change. 8. This would be consistent with suggestions that in sub-Saharan Africa numbers could have dropped at times as low as 2000, for perhaps as long as 100,000 years, before numbers began to expand again in the late Stone Age. 18. Other animals. European bison, also called wisent bison bonosus, faced extinction in the early 20th century. The animals living today are all descended from 12 individuals and they have extremely low genetic variation, which may be beginning to affect the reproductive ability of bulls. 19. The population of American bison fell due to overhunting, nearly leading to extinction around the year 1890, though it has since begun to recover. See table. Overhunting pushed the northern elephant seal to the brink of extinction by the late 19th century. Although they have made a comeback, the genetic variation within the population remains very low. A classic example of a population bottleneck is that of the northern elephant seal, whose population fell to about 30 in the 1890s. Although it now numbers in the hundreds of thousands, the potential for bottlenecks within colonies remains. Dominant bulls are able to mate with the largest number of females, sometimes as many as 100. With so much a colony's offspring descended from just one dominant male, genetic diversity is limited, making the species more vulnerable to diseases and genetic mutations. The golden hamster is a similarly bottlenecked species, with the vast majority of domesticated hamsters descended from a single litter found in the Syrian desert around 1930, and very few wild golden hamsters remain. An extreme example of a population bottleneck is the New Zealand black robin, of which every specimen today is a descendant of a single female, called Old Blue. The black robin population is still recovering from its low point of only 5 individuals in 1980. The genome of the giant panda shows evidence of a severe bottleneck about 43,000 years ago. 20. There is also evidence of at least one primate species, the golden snub-nosed monkey, that also suffered from a bottleneck around this time. An unknown environmental event is suspected to have caused the bottlenecks observed in both of these species. The bottlenecks likely cause the low genetic diversity observed in both species. Other facts can sometimes be inferred from an observed population bottleneck. Among the Galapagos Islands giant tortoises, themselves a prime example of a bottleneck, the comparatively large population on the slopes of the Alcedo volcano is significantly less diverse than 4 other tortoise populations on the same island. Dominant bulls are able to mate with the largest number of females, sometimes as many as 100. With so much a colony's offspring descended from just one dominant male, genetic diversity is limited, making the species more vulnerable to diseases and genetic mutations. The golden hamster is a similarly bottlenecked species, with the vast majority of domesticated hamsters descended from a single litter found in the Syrian desert around 1930, and very few wild golden hamsters remain. An extreme example of a population bottleneck is the New Zealand black robin, of which every specimen today is a descendant of a single female, called Old Blue. The black robin population is still recovering from its low point of only 5 individuals in 1980. The genome of the giant panda shows evidence of a severe bottleneck about 43,000 years ago. 20. There is also evidence of at least one primate species, the golden snub-nosed monkey, that also suffered from a bottleneck around this time. An unknown environmental event is suspected to have caused the bottlenecks observed in both of these species. The bottlenecks likely cause the low genetic diversity observed in both species. Other facts can sometimes be inferred from an observed population bottleneck. Among the Galapagos Islands giant tortoises, themselves a prime example of a bottleneck, the comparatively large population on the slopes of the Alcedo volcano is significantly less diverse than 4 other tortoise populations on the same island. DNA analyses date the bottleneck to around 88,000 years before present YBP. 21. About 100,000 YBP the volcano erupted violently, deeply burying much of the tortoise habitat in pumice and ash. Another example can be seen in the greater prairie chickens, which were prevalent in North America until the 20th century. In Illinois alone, the number of greater prairie chickens plummeted from over 100 million in 1900 to about 46 in 1998. 22. These declines in population were the result of hunting and habitat destruction, but the random consequences have also caused a great loss in species diversity. DNA analysis comparing the birds from 1990 and mid-century shows a steep genetic decline in recent decades. Management of the greater prairie chickens now includes genetic rescue efforts including the translocation prairie chickens between leks to increase each population's genetic diversity. 22. Population bottlenecking poses a major threat to the stability of species populations as well. Papilio homerus is the largest butterfly in the Americas and is endangered according to the IUCN. The disappearance of a central population poses a major threat of population bottleneck. The remaining 2 populations are now geographically isolated and the populations face an unstable future with limited remaining opportunity for gene flow. 23. Genetic bottlenecks exist in cheetahs. 24-25. Selective breeding. Bottlenecks also exist among purebred animals, e.g., dogs and cats, pugs, Persian, because breeders limit their gene pools by a few, show-winning individuals for their looks and behaviors. The extensive use of desirable individual animals at the exclusion of others can result in a popular sire effect. Selective breeding for dog breeds cause constricting breed-specific bottlenecks. 26. These bottlenecks have led to dogs having an average of 2–3% more genetic loading than gray wolves. 27. The strict breeding programs and population bottlenecks have led to the prevalence of diseases such as heart disease, blindness, cancers, hip dysplasia, and cataracts. 26. Selective breeding to produce high-yielding crops has caused genetic bottlenecks in these crops and has led to genetic homogeneity. 28. This reduced genetic diversity in many crops could lead to broader susceptibility to new diseases or pests, which threatens global food security. 29. Plants. Research showed that there is incredibly low, nearly undetectable amounts of genetic diversity in the genome of the Wollemi pine, Wollemia nobilis. 30. The IUCN found a population count of 80 mature individuals and about 300 seedlings and juveniles in 2011, and previously, the Wollemi pine had fewer than 50 individuals in the wild. 31. The low population size and low genetic diversity indicates that the Wollemi pine went through a severe population bottleneck. A population bottleneck was created in the 1970s through the conservation efforts of the endangered Mauna Kea silversword, Argyroxiphium sandwichense ssp. Sandwichense. 32. The small natural population of silversword was augmented through the 1970s with outplanted individuals. All of the outplanted silversword plants were found to be first or subsequent generation offspring of just two maternal founders. The low amount of polymorphic loci in the outplanted individuals led to the population bottleneck, causing the loss of the marker allele at 8 of the loci. See also. Population genetics. Population genetics is a subfield of genetics that deals with genetic differences within and among populations, and is a part of evolutionary biology. Studies in this branch of biology examine such phenomena as adaptation, speciation, and population structure. 1. Population genetics was a vital ingredient in the emergence of the modern evolutionary synthesis. Its primary founders were Sewall Wright, J.B.S. Haldane and Ronald Fisher, who also laid the foundations for the related discipline of quantitative genetics. Traditionally a highly mathematical discipline, modern population genetics encompasses theoretical, laboratory, and fieldwork. Population genetic models are used both for statistical inference from DNA sequence data and for proof-disproof of concept. 2. What sets population genetics apart from newer, more phenotypic approaches to modeling evolution, such as evolutionary game theory and adaptive dynamics, is its emphasis on such genetic phenomena as dominance, epistasis, the degree to which genetic recombination breaks linkage disequilibrium, and the random phenomena of mutation and genetic drift. This makes it appropriate for comparison to population genomics data. History. Population genetics began as a reconciliation of Mendelian inheritance and biostatistics models. Natural selection will only cause evolution if there is enough genetic variation in a population. Before the discovery of Mendelian genetics, one common hypothesis was blending inheritance. But with blending inheritance, genetic variance would be rapidly lost, making evolution by natural or sexual selection implausible. The Hardy-Weinberg principle provides the solution to how variation is maintained in a population with Mendelian inheritance. According to this principle, the frequencies of alleles variations in a gene will remain constant in the absence of selection, mutation, migration and genetic drift. 3. The typical white-bodied form of the peppered moth. Industrial melanism, the black-bodied form of the peppered moth appeared in polluted areas. The next key step was the work of the British biologist and statistician Ronald Fisher. In a series of papers starting in 1918 and culminating in his 1930 book The Genetical Theory of Natural Selection, Fisher showed that the continuous variation measured by the biometricians could be produced by the combined action of many discrete genes, and that natural selection could change allele frequencies in a population, resulting in evolution. In a series of papers beginning in 1924, another British geneticist, J.B.S. Haldane, worked out the mathematics of allele frequency change at a single genelocus under a broad range of conditions. Haldane also applied statistical analysis to real-world examples of natural selection, such as peppered moth evolution and industrial melanism, and showed that selection coefficients could be larger than Fisher assumed, leading to more rapid adaptive evolution as a camouflage strategy following increased pollution. 4. 5. The American biologist Sewall Wright, who had a background in animal breeding experiments, focused on combinations of interacting genes, and the effects of inbreeding on small, relatively isolated populations that exhibited genetic drift. In 1932 Wright introduced the concept of an adaptive landscape and argued that genetic drift and inbreeding could drive a small, isolated subpopulation away from an adaptive peak, allowing natural selection to drive it towards different adaptive peaks. The work of Fisher, Haldane and Wright founded the discipline of population genetics. This integrated natural selection with Mendelian genetics, which was the critical first step in developing a unified theory of how evolution worked. 4. 5. John Maynard Smith was Haldane's pupil, whilst W. D. Hamilton was influenced by the writings of Fisher. The American George R. Price worked with both Hamilton and Maynard Smith. American Richard Lewontin and Japanese Moto Kimura were influenced by Wright and Haldane. Modern synthesis. The mathematics of population genetics were originally developed as the beginning of the modern synthesis. Authors such as Beatty, 6, have asserted that population genetics defines the core of the modern synthesis. For the first few decades of the 20th century, most field naturalists continued to believe that Lamarckism and orthogenesis provided the best explanation for the complexity they observed in the living world. 7. During the modern synthesis, these ideas were purged, and only evolutionary causes that could be expressed in the mathematical framework of population genetics were retained. 8. Consensus was reached as to which evolutionary factors might influence evolution, but not as to the relative importance of the various factors. 8. Theodosius Dobzhansky, a postdoctoral worker in T. H. Morgan's lab, had been influenced by the work on genetic diversity by Russian geneticists such as Sergei Chetverikov. He helped to bridge the divide between the foundations of microevolution developed by the population geneticists and the patterns of macroevolution observed by field biologists, with his 1937 book Genetics and the Origin of Species. Dobzhansky examined the genetic diversity of wild populations and showed that, contrary to the assumptions of the population geneticists, these populations had large amounts of genetic diversity, with marked differences between subpopulations. The book also took the highly mathematical work of the population geneticists and put it into a more accessible form. Many more biologists were influenced by population genetics via Dobzhansky than were able to read the highly mathematical works in the original. 9. In Great Britain, E. B. Ford, the pioneer of ecological genetics, continued throughout the 1930s and 1940s to empirically demonstrate the power of selection due to ecological factors including the ability to maintain genetic diversity through genetic polymorphisms such as human blood types. Ford's work, in collaboration with Fisher, contributed to a shift in emphasis during the modern synthesis towards natural selection as the dominant force. 4, 5, 11, 12. Neutral theory and origin fixation dynamics. The original, modern synthesis view of population genetics assumes that mutations provide ample raw material, and focuses only on the change in frequency of alleles within populations. 13. The main processes influencing allele frequencies are natural selection, genetic drift, 4, 5, 11, 12. Neutral theory and origin fixation dynamics. The original, modern synthesis view of population genetics assumes that mutations provide ample raw material, and focuses only on the change in frequency of alleles within populations. 13. The main processes influencing allele frequencies are natural selection, genetic drift, gene flow and recurrent mutation. Fisher and Wright had some fundamental disagreements about the relative roles of selection and drift. 14. The availability of molecular data on all genetic differences led to the neutral theory of molecular evolution. In this view, many mutations are deleterious and so never observed, and most of the remainder are neutral, i.e. are not under selection. With the fate of each neutral mutation left to chance genetic drift, the direction of evolutionary change is driven by which mutations occur, and so cannot be captured by models of change in the frequency of existing alleles alone. 13, 15. The origin fixation view of population genetics generalizes this approach beyond strictly neutral mutations, and sees the rate at which a particular change happens as the product of the mutation rate and the fixation probability. 13. 4 processes. Selection. Natural selection, which includes sexual selection, is the fact that some traits make it more likely for an organism to survive and reproduce. Population genetics describes natural selection by defining fitness as a propensity or probability of survival and reproduction in a particular environment. The fitness is normally given by the symbol W equals 1s, where s is the selection coefficient. Natural selection acts on phenotypes, so population genetic models assume relatively simple relationships to predict the phenotype and hence fitness from the allele at one or a small number of loci. In this way, natural selection converts differences in the fitness of individuals with different phenotypes into changes in allele frequency in a population over successive generations. Before the advent of population genetics, many biologists doubted that small differences in fitness were sufficient to make a large difference to evolution. 9. Population geneticists addressed this concern in part by comparing selection to genetic drift. Selection can overcome genetic drift when S is greater than 1 divided by the effective population size. When this criterion is met, the probability that a new advantageous mutant becomes fixed is approximately equal to 2's. 16, 17. The time until fixation of such an allele is approximately 18. Dominance. Dominance means that the phenotypic and/or fitness effect of one allele at a locus depends on which allele is present in the second copy for that locus. Consider 3 genotypes at one locus, with the following fitness values: 19. Genotype: A1 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 A2 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 relative fitness: 11 HS 1S. S is the selection coefficient and H is the dominance coefficient. The value of H yields the following information. H equals O A1 dominant, A2 recessive H equals 1 A2 dominant, A1 recessive 0 less than H less than 1 incomplete dominance H less than 0 overdominance. H greater than 1 underdominance. Epistasis. The logarithm of fitness is a function of the number of deleterious mutations. Synergistic epistasis is represented by the red line. Each subsequent deleterious mutation has a larger proportionate effect on the organism's fitness. Antagonistic epistasis is in blue. The black line shows the non-epistatic case, where fitness is the product of the contributions from each of its loci. Epistasis means that the phenotypic and/or fitness effect of an allele at one locus depends on which alleles are present at other loci. Selection does not act on a single locus, but on a phenotype that arises through development from a complete genotype. 20. However, many population genetics models of sexual species are single locus models, where the fitness of an individual is calculated as the product of the contributions from each of its loci, effectively assuming no epistasis. In fact, the genotype to fitness landscape is more complex. Population genetics must either model this complexity in detail, or capture it by some simpler average rule. Empirically, beneficial mutations tend to have a smaller fitness benefit when added to a genetic background that already has high fitness. This is known as diminishing returns epistasis. 21. When deleterious mutations also have a smaller fitness effect on high fitness backgrounds, this is known as synergistic epistasis. However, the effect of deleterious mutations tends on average to be very close to multiplicative, or can even show the opposite pattern, known as antagonistic epistasis. 22. Population genetics glossary. Species, a group of closely related organisms which, if sexual, are capable of interbreeding and producing fertile offspring. Population, the set of individuals of a particular species in a given area. Gene pool, the collective genetic information contained within a population of sexually reproducing organisms, ignores linkage disequilibrium. Allele frequency, the frequency or proportion of a particular allele of a gene within a population. Synergistic epistasis is central to some theories of the purging of mutation load, 23, and to the evolution of sexual reproduction. Mutation. Drosophila melanogaster. The genetic process of mutation takes place within an individual, resulting in heritable changes to the genetic material. This process is often characterized by a description of the starting and ending states, or the kind of change that has happened at the level of DNA, e.g. A T to C mutation, a 1BP deletion of genes or proteins, e.g., a null mutation, a loss of function mutation, or at a higher phenotypic level, e.g., red eye mutation. Single nucleotide changes are frequently the most common type of mutation, but many other types of mutation are possible, and they occur at widely varying rates that may show systematic asymmetries or biases. Mutation bias. Mutations can involve large sections of DNA becoming duplicated, usually through genetic recombination. 24. This leads to copy number variation within a population. Duplications are a major source of raw material for evolving new genes. 25. Other types of mutation occasionally create new genes from previously non-coding DNA. 26. 27. In the distribution of fitness effects DFE, for new mutations, only a minority of mutations are beneficial. Mutations with gross effects are typically deleterious. Studies in the fly Drosophila melanogaster suggest that if a mutation changes a protein produced by a gene, this will probably be harmful, with about 70% of these mutations having damaging effects, and the remainder being either neutral or weakly beneficial. 28. This biological process of mutation is represented in population genetic models in one of two ways, either as a deterministic pressure of recurrent mutation on allele frequencies, or a source of variation. In deterministic theory, evolution begins with a predetermined set of alleles and proceeds by shifts in continuous frequencies, as if the population is infinite. The occurrence of mutations in individuals is represented by a population level, force, or pressure, of mutation, i.e., the force of innumerable events of mutation with a scaled magnitude U applied to shifting frequencies FA1 to FA2. For instance, in the classic mutation selection balance model, 29, the force of mutation pressure pushes the frequency of an allele upward, and selection against its deleterious effects pushes the frequency downward, so that a balance is reached at equilibrium, given, in the simplest case, by F equals US. This concept of mutation pressure is mostly useful for considering the implications of deleterious mutation, such as the mutation load and its implications for the evolution of the mutation rate. 30. Transformation of populations by mutation pressure is unlikely. Haldane, 31, argued that it would require high mutation rates unopposed by selection, and Kimura, 32, concluded even more pessimistically that even this was unlikely, as the process would take too long. See evolution by mutation pressure. However, evolution by mutation pressure is possible under some circumstances and has long been suggested as a possible cause for the loss of unused traits. 33. For example, pigments are no longer useful when animals live in the darkness of caves, and tend to be lost. 34. An experimental example involves the loss of sporulation in experimental populations of B. subtilis. Sporulation is a complex trait encoded by many loci, such that the mutation rate for loss of the trait was estimated as an unusually high value. 35. Loss of sporulation in this case can occur by recurrent mutation, without requiring selection for the loss of sporulation ability. When there is no selection for loss of function, the speed at which loss evolves depends more on the mutation rate than it does on the effective population size. 36. Indicating that it is driven more by mutation than by genetic drift. The role of mutation as a source of novelty is different from these classical models of mutation pressure. When population genetic models include a rate-dependent process of mutational introduction or origination, i.e., a process that introduces new alleles including neutral and beneficial ones, then the properties of mutation may have a more direct impact on the rate and direction of evolution, even if the rate of mutation is very low. 37, 38. That is, the spectrum of mutation may become very important, particularly mutation biases, predictable differences in the rates of occurrence for different types of mutations, because bias in the introduction of variation can impose biases on the course of evolution. 39. Mutation plays a key role in other classical and recent theories including Muller's ratchet, subfunctionalization, Eigen's concept of an error catastrophe and Lynch's mutational hazard hypothesis. Genetic drift. Genetic drift is a change in allele frequencies caused by random sampling. 40. That is, the alleles in the offspring are a random sample of those in the parents. 41. Genetic drift may cause gene variants to disappear completely, and thereby reduce genetic variability. In contrast to natural selection, which makes gene variants more common or less common depending on their reproductive success, 42, the changes due to genetic drift are not driven by environmental or adaptive pressures, and are equally likely to make an allele more common as less common. The effect of genetic drift is larger for alleles present in few copies than when an allele is present in many copies. The population genetics of genetic drift are described using either branching processes or a diffusion equation describing changes in allele frequency. 43. These approaches are usually applied to the Wright, Fisher, and Moran models of population genetics. Assuming genetic drift is the only evolutionary force acting on an allele, after T generations in many replicated populations, starting with allele frequencies of P and Q, the variance in allele frequency across those populations is 44. Ronald Fisher held the view that genetic drift plays at the most a minor role in evolution, and this remained the dominant view for several decades. No population genetics perspective have ever given genetic drift a central role by itself, but some have made genetic drift important in combination with another non-selective force. The shifting balance theory of Sewall Wright held that the combination of population structure and genetic drift was important. Moto Kimura's neutral theory of molecular evolution claims that most genetic differences within and between populations are caused by the combination of neutral mutations and genetic drift. 45. The role of genetic drift by means of sampling error in evolution has been criticized by John H. Gillespie, 46, and Will Provini, 47, who argue that selection on linked sites is a more important stochastic force, doing the work traditionally ascribed to genetic drift by means of sampling error. The mathematical properties of genetic draft are different from those of genetic drift. 48. The direction of the random change in allele frequency is autocorrelated across generations. 40. Gene flow. Gene flow is the transfer of alleles from one population to another population through immigration of individuals. In this example, one of the birds from population A immigrates to population B, which has fewer of the dominant alleles, and through mating incorporates its alleles into the other population. Because of physical barriers to migration, along with the limited tendency for individuals to move or spread, the agility, and tendency to remain or come back to natal place, phylopatry, natural populations rarely all interbreed as may be assumed in theoretical random models panmixy. 49. There is usually a geographic range within which individuals are more closely related to one another than those randomly selected from the general population. This is described as the extent to which a population is genetically structured. 50. The Great Wall of China is an obstacle to gene flow of some terrestrial species. 51. Genetic structuring can be caused by migration due to historical climate change, species range expansion or current availability of habitat. Gene flow is hindered by mountain ranges, oceans and deserts or even human-made structures such as the Great Wall of China, which has hindered the flow of plant genes. 51. Gene flow is the exchange of genes between populations or species, breaking down the structure. Examples of gene flow within a species include the migration and then breeding of organisms, or the exchange of pollen. Gene transfer between species includes the formation of hybrid organisms and horizontal gene transfer. Population genetic models can be used to identify which populations show significant genetic isolation from one another, and to reconstruct their history. 52. Subjecting a population to isolation leads to inbreeding depression. Migration into a population can introduce new genetic variants. 53. Potentially contributing to evolutionary rescue. If a significant proportion of individuals or gametes migrate, it can also change allele frequencies, e.g. giving rise to migration load. 54. In the presence of gene flow, other barriers to hybridization between two diverging populations of an outcrossing species are required for the populations to become new species. Horizontal gene transfer. Current tree of life showing vertical and horizontal gene transfers. Horizontal gene transfer is the transfer of genetic material from one organism to another organism that is not its offspring. This is most common among prokaryotes. 55. In medicine, this contributes to the spread of antibiotic resistance, as when one bacteria acquires resistance genes it can rapidly transfer them to other species. 56. Horizontal transfer of genes from bacteria to eukaryotes such as the yeast Saccharomyces cerevisiae and the Adzuki bean beetle Calosobruchus chinensis may also have occurred. 57. 58. An example of larger-scale transfers are the eukaryotic bdelloid rotifers, which appear to have received a range of genes from bacteria, fungi, and plants. 59. Viruses can also carry DNA between organisms, allowing transfer of genes even across biological domains. 60. Large-scale gene transfer has also occurred between the ancestors of eukaryotic cells and prokaryotes, during the acquisition of chloroplasts and mitochondria. 61. Linkage. If all genes are in linkage equilibrium, the effect of an allele at one locus can be averaged across the gene pool at other loci. In reality, one allele is frequently found in linkage disequilibrium with genes at other loci, especially with genes located nearby on the same chromosome. Recombination breaks up this linkage disequilibrium too slowly to avoid genetic hitchhiking, where an allele at one locus rises to high frequency because it is linked to an allele under selection at a nearby locus. Linkage also slows down the rate of adaptation, even in sexual populations. 62, 63, 64. The effect of linkage disequilibrium in slowing down the rate of adaptive evolution arises from a combination of the Hill-Robertson effect, delays in bringing beneficial mutations together, and background selection, delays in separating beneficial mutations from deleterious hitchhikers. Linkage is a problem for population genetic models that treat one gene locus at a time. It can, however, be exploited as a method for detecting the action of natural selection via selective sweeps. In the extreme case of an asexual population, linkage is complete, and population genetic equations can be derived and solved in terms of a traveling wave of genotype frequencies along a simple fitness landscape. 65. Most microbes, such as bacteria, are asexual. The population genetics of their adaptation have two contrasting regimes. When the product of the beneficial mutation rate and population size is small, asexual populations follow a successional regime of origin fixation dynamics, with adaptation rates strongly dependent on this product. When the product is much larger, asexual populations follow a concurrent mutations regime with adaptation rate less dependent on the product, characterized by clonal interference and the appearance of a new beneficial mutation before the last one has fixed. Applications. Explaining levels of genetic variation. Neutral theory predicts that the level of nucleotide diversity in a population will be proportional to the product of the population size and the neutral mutation rate. The fact that levels of genetic diversity vary much less than population size is due is known as the paradox of variation. 66. While high level neutral theory predicts that the level of nucleotide diversity in a population will be proportional to the product of the population size and the neutral mutation rate. The fact that levels of genetic diversity vary much less than population size is due is known as the paradox of variation. 66. While high levels of genetic diversity were one of the original arguments in favor of neutral theory, the paradox of variation has been one of the strongest arguments against neutral theory. It is clear that levels of genetic diversity vary greatly within a species as a function of local recombination rate, due to both genetic hitchhiking and background selection. Most current solutions to the paradox of variation invoke some level of selection at linked sites. 67. For example, one analysis suggests that larger populations have more selective sweeps, which remove more neutral genetic diversity. 68. A negative correlation between mutation rate and population size may also contribute. 69. Life history affects genetic diversity more than population history does, e.g. our strategists have more genetic diversity. 67. Detecting selection. Population genetics models are used to infer which genes are undergoing selection. One common approach is to look for regions of high linkage disequilibrium and low genetic variance along the chromosome, to detect recent selective sweeps. A second common approach is the McDonald-Kreitman test, which compares the amount of variation within a species' polymorphism to the divergence between species substitutions at two types of sites, one assumed to be neutral. Typically, synonymous sites are assumed to be neutral. 70. Genes undergoing positive selection have an excess of divergent sites relative to polymorphic sites. The test can also be used to obtain a genome-wide estimate of the proportion of substitutions that are fixed by positive selection, alpha. 71. 72. According to the neutral theory of molecular evolution, this number should be near zero. High numbers have therefore been interpreted as a genome-wide falsification of neutral theory. 73. Demographic inference. The simplest test for population structure in a sexually reproducing, diploid species, is to see whether genotype frequencies follow Hardy-Weinberg proportions as a function of allele frequencies. For example, in the simplest case of a single locus with two alleles denoted A and at frequencies P and Q, random mating predicts freq A equals P2 for the AA homozygotes, freq A equals Q2 for the A homozygotes, and freq A equals 2PQ for the heterozygotes. In the absence of population structure, Hardy-Weinberg proportions are reached within 1 to 2 generations of random mating. More typically, there is an excess of homozygotes, indicative of population structure. The extent of this excess can be quantified as the inbreeding coefficient, F. Individuals can be clustered into K subpopulations. 74. 75. The degree of population structure can then be calculated using FST, which is a measure of the proportion of genetic variance that can be explained by population structure. Genetic population structure can then be related to geographic structure, and genetic admixture can be detected. Coalescent theory relates genetic diversity in a sample to demographic history of the population from which it was taken. It normally assumes neutrality, and so sequences from more neutrally evolving portions of genomes are therefore selected for such analyses. It can be used to infer the relationships between species phylogenetics as well as the population structure, demographic history e.g. population bottlenecks, population growth, biological dispersal, source sink dynamics 76, and introgression within a species. Another approach to demographic inference relies on the allele frequency spectrum. 77. Evolution of genetic systems. By assuming that there are loci that control the genetic system itself, population genetic models are created to describe the evolution of dominance and other forms of robustness, the evolution of sexual reproduction and recombination rates, the evolution of mutation rates, the evolution of evolutionary capacitors, the evolution of costly signaling traits, the evolution of aging, and the evolution of cooperation. For example, most mutations are deleterious, so the optimal mutation rate for a species may be a trade-off between the damage from a high deleterious mutation rate and the metabolic costs of maintaining systems to reduce the mutation rate, such as DNA repair enzymes. 78. One important aspect of such models is that selection is only strong enough to purge deleterious mutations and hence overpower mutational bias towards degradation if the selection coefficient S is greater than the inverse of the effective population size. This is known as the drift barrier and is related to the nearly neutral theory of molecular evolution. Drift barrier theory predicts that species with large effective population sizes will have highly streamlined, efficient genetic systems, while those with small population sizes will have bloated and complex genomes containing, for example, introns and transposable elements. 79. However, somewhat paradoxically, species with large population sizes might be so tolerant to the consequences of certain types of errors that they evolve higher error rates, e.g. in transcription and translation, than small populations. 80. See also. Allele frequency spectrum. Quantitative genetics. Quantitative genetics is the study of quantitative traits, which are phenotypes that vary continuously, such as height or mass, as opposed to phenotypes and gene products that are discretely identifiable, such as eye color, or the presence of a particular biochemical. Both of these branches of genetics use the frequencies of different alleles of a gene in breeding populations, gamodemes, and combine them with concepts from simple Mendelian inheritance to analyze inheritance patterns across generations and descendant lines. While population genetics can focus on particular genes and their subsequent metabolic products, quantitative genetics focuses more on the outward phenotypes, and makes only summaries of the underlying genetics. Due to the continuous distribution of phenotypic values, quantitative genetics must employ many other statistical methods, such as the effect size, the mean and the variance to link phenotypes attributes to genotypes. Some phenotypes may be analyzed either as discrete categories or as continuous phenotypes, depending on the definition of cutoff points, or on the metric used to quantify them. 1:27-69 Mendel himself had to discuss this matter in his famous paper, 2, especially with respect to his P's attribute tall dwarf, which actually was derived by adding a cutoff point to length of stem. 3-4 Analysis of quantitative trait loci, or QTLs, 5-6-7, is a more recent addition to quantitative genetics, linking it more directly to molecular genetics. Gene effects. In diploid organisms, the average genotypic value, locus value, may be defined by the allele effect, together with a dominance effect, and also by how genes interact with genes at other loci, epistasis. The founder of quantitative genetics, Sir Ronald Fisher, perceived much of this when he proposed the first mathematics of this branch of genetics. 8. Gene effects and phenotype values. Being a statistician, he defined the gene effects as deviations from a central value, enabling the use of statistical concepts such as mean and variance, which use this idea. 9. The central value he chose for the gene was the midpoint between the two opposing homozygotes at the one locus. The deviation from there to the greater homozygous genotype can be named plus a, and therefore it is a, from that same midpoint to the lesser homozygote genotype. This is the allele effect mentioned above. The heterozygote deviation from the same midpoint can be named d, this being the dominance effect referred to above. 10th, the diagram depicts the idea. However, in reality we measure phenotypes, and the figure also shows how observed phenotypes relate to the gene effects. Formal definitions of these effects recognize this phenotypic focus. 11-12 Epistasis has been approached statistically as interaction, i.e., inconsistencies. 13, but epigenetics suggests a new approach may be needed. If 0 less than d less than a, the dominance is regarded as partial or incomplete, while d equals a indicates full or classical dominance. Previously, d greater than a was known as over-dominance. 14. Mendel's P attribute, length of stem, provides us with a good example. 3. Mendel stated that the tall true breeding parents ranged from 6 to 7 feet in stem length, 183-213 centimeters, giving a median of 198 centimeters equals P1. The short parents ranged from 0.75 to 1.25 feet in stem length, 23-46 centimeters, with a rounded median of 34 centimeters equals P2. Their hybrid ranged from 6-7.5 feet in length, 183-229 centimeters, with a median of 206 centimeters equals F1. The mean of P1 and P2 is 116 centimeters, this being the phenotypic value of the homozygote's midpoint, MP. The allele affect A is P1 MP equals 82 centimeters equals P2 MP. The dominance effect D is F1 MP equals 90 centimeters. 15. This historical example illustrates clearly how phenotype values and gene effects are linked. Allele and genotype frequencies. To obtain means, variances and other statistics, both quantities and their occurrences are required. The gene effects above provide the framework for quantities, and the frequencies of the contrasting alleles in the fertilization gamete pool provide the information on occurrences. Analysis of sexual reproduction. Commonly, the frequency of the allele causing more in the phenotype, including dominance, is given the symbol P, while the frequency of the contrasting allele is Q. An initial assumption made when establishing the algebra was that the parental population was infinite and random mating, which was made simply to facilitate the derivation. The subsequent mathematical development also implied that the frequency distribution within the effective gamete pool was uniform. There were no local perturbations where P and Q varied. Looking at the diagrammatic analysis of sexual reproduction, this is the same as declaring that PP equals PG equals P, and similarly for Q. 14. This mating system, dependent upon these assumptions, became known as panmixia. Panmixia rarely actually occurs in nature. 16. 152 to 180. 17. As gamete distribution may be limited, for example by dispersal restrictions or by behavior, or by chance sampling those local perturbations mentioned above. It is well known that there is a huge wastage of gametes in nature, which is why the diagram depicts a potential gamete pool separately to the actual gamete pool. Only the latter sets the definitive frequencies for the zygotes. This is the true gamodeme. Gamo refers to the gametes, and deme derives from Greek for population. But, under Fisher's assumptions, the gamodeme can be effectively extended back to the potential gamete pool, and even back to the parental base population, the source population. The random sampling arising when small, actual, gamete pools are sampled from a large, potential, gamete pool is known as genetic drift, and is considered subsequently. While panmixia may not be widely extant, the potential for it does occur, although it may be only ephemeral because of those local perturbations. It has been shown, for example, that the F2 derived from random fertilization of F1 individuals and allogamous F2 following hybridization is an origin of a new potentially panmictic population. 19. It has also been shown that if panmictic random fertilization occurred continually, it would maintain the same allele and genotype frequencies across each successive panmictic sexual generation, this being the Hardy-Weinberg equilibrium. 34-39, 2021, 22, 23. However, as soon as genetic drift was initiated by local random sampling of gametes, the equilibrium would cease. Random fertilization. Male and female gametes within the actual fertilizing pool are considered usually to have the same frequencies for their corresponding alleles. Exceptions have been considered. This means that when P male gametes carrying the A allele randomly fertilize P female gametes carrying that same allele, the resulting zygote has genotype A, and, under random fertilization, the combination occurs with a frequency of PXP equals P2. Similarly, the zygote A occurs with a frequency of Q2. Heterozygotes A can arise in two ways, when P male allele randomly fertilize Q female allele gametes, and vice versa. The resulting frequency for the heterozygous zygotes is thus 2pq. 1332. Notice that such a population is never more than half heterozygous, this maximum occurring when P equals Q equals 0.5. In summary then, under random fertilization, the zygote genotype frequencies are the quadratic expansion of the gametic allelic frequencies. The equals 1 inch states that the frequencies are in fraction form, not percentages, and that there are no omissions within the framework proposed. Notice that random fertilization and panmixia are not synonyms. Mendel's research cross, a contrast. Mendel's P experiments were constructed by establishing true breeding parents with opposite phenotypes for each attribute. This meant that each opposite parent was homozygous for its respective allele only. In our example, tall versus dwarf, the tall parent would be genotype TT with P equals 1 and Q equals 0, while the dwarf parent would be genotype TT with Q equals 1 and P equals 0. After controlled crossing, their hybrid is TT, with P equals Q equals 1. 2. However, the frequency of this heterozygote equals 1, because this is the F1 of an artificial cross, it has not arisen through random fertilization. 24. The F2 generation was produced by natural self-pollination of the F1 with monitoring against insect contamination, resulting in P equals Q equals 1. 2 being maintained. Such an F2 is said to be autogamous. However, the genotype frequencies 0.25 tt, 0.5 tt, 0.25 tt have arisen through a mating system very different from random fertilization, and therefore the use of the quadratic expansion has been avoided. The numerical values obtained were the same as those for random fertilization only because this is a special case of having originally crossed homozygous opposite parents. 25. We can notice that, because of the dominance of T frequency 0.25 plus 0.5 over TT frequency 0.25, the 3:1 ratio is still obtained. A cross such as Mendel's, where true breeding largely homozygous opposite parents are crossed in a controlled way to produce an F1, is a special case of hybrid structure. The F1 is often regarded as entirely heterozygous for the gene under consideration. However, this is an oversimplification and does not apply generally, for example when individual parents are not homozygous, or when populations interhybridize to form hybrid swarms. 24. The general properties of intraspecies hybrids F1 and F2 both, autogamous and allogamous, are considered in a later section. Self-fertilization, an alternative. Having noticed that the P is naturally self-pollinated, we cannot continue to use it as an example for illustrating random fertilization properties. Self-fertilization, selfing, is a major alternative to random fertilization, especially within plants. Most of the Earth's cereals are naturally self-pollinated: rice, wheat, barley, for example, as well as the pulses. Considering the millions of individuals of each of these on Earth at any time, it is obvious that self-fertilization is at least as significant as random fertilization. Self-fertilization is the most intensive form of inbreeding, which arises whenever there is restricted independence in the genetical origins of gametes. Such reduction in independence arises if parents are already related, and/or from genetic drift or other spatial restrictions on gamete dispersal. Path analysis demonstrates that these are tantamount to the same thing. 26-27. Arising from this background, the inbreeding coefficient, often symbolized as F or F, quantifies the effect of inbreeding from whatever cause. There are several formal definitions of F, and some of these are considered in later sections. For the present, note that for a long-term self-fertilized species F equals 1. Natural self-fertilized populations are not single, pure lines, however, but mixtures of such lines. This becomes particularly obvious when considering more than one gene at a time. Therefore, allele frequencies P and Q other than 1 or 0 are still relevant in these cases. Refer back to the Mendel cross-section. The genotype frequencies take a different form, however. In general, the genotype frequencies become for A and for A and for A. 1365. Notice that the frequency of the heterozygote declines in proportion to F. When F equals 1, these three frequencies become respectively P, 0 and Q. Conversely, when F equals 0, they reduce to the random fertilization quadratic expansion shown previously. Population mean. The population mean shifts the central reference point from the homozygote midpoint, MP, to the mean of a sexually reproduced population. This is important not only to relocate the focus into the natural world, but also to use a measure of central tendency used by statistics biometrics. In particular, the square of this mean is the correction factor, which is used to obtain the genotypic variances later. 9. Population mean across all values of p, for various d effects. For each genotype in turn, its allele effect is multiplied by its genotype frequency, and the products are accumulated across all genotypes in the model. Some algebraic